In this video, we will talk about nutrients beyond absorption and in particular about the metabolism of macronutrients. The main nutrients in food are carbohydrates, proteins, and lipids, and these nutrients are considered macronutrients because they are our main source of energy. The digestion of food begins in the oral cavity. From the oral cavity, the food bolus passes to the esophagus, stomach, and small intestine, where, thanks to bile and enzymes, takes place the final digestion of the macronutrients present in food. They are broken down to the biomolecules that compose them, which are monosaccharides, mainly glucose, and carbohydrates, amino acids and proteins, and fatty acids and glycerol and lipids. And as the last step, these biomolecules are absorbed. Once glucose has been absorbed and passed into the blood, it goes to the liver where it can be used for energy or stored. Glucose can also go directly to adipose tissue where it is stored as fat, and to other tissues such as muscle where it can be used for energy or stored, or to the brain where it is only used for energy. In fact, the brain is very dependent on blood glucose levels since glucose is practically its only source of energy and it cannot store it. Once absorbed, amino acids also pass into the blood and go to the liver where they are stored as proteins. And finally, once the fatty acids and glycerol are absorbed and pass into the blood, they also go to the liver where they participate in the formation of lipoproteins. These lipoproteins pass into the blood and go to organs that use fatty acids as a source of energy, such as the heart, and also to adipose tissue where fatty acids and glycerol are stored as fat. Fatty acids and glycerol can also go directly to adipose tissue for storage or to tissues that can use them as an energy source. It is important that blood glucose levels remain stable, and for this, there is a control system in which the pancreas plays a crucial role. When blood glucose levels increase after eating, the pancreas detects it and produces a hormone called insulin, the function of which is to facilitate the entry and storage of the different biomolecules, particularly glucose, in the liver and tissues. After ingestion, glucose is stored in the liver and muscle, protein in the liver, and fat in adipose tissue. If we do a short fasting for less than 12 hours, that is the usual fasting between meals, blood glucose levels begin to drop. This decrease is detected by the liver, which begins to mobilize the glucose it has stored and releases it into the blood to maintain stable blood glucose levels. This glucose can then go to organs such as the brain that needs it to function. Other tissues that have stored glucose, such as muscle, can use their own stored glucose. If the fasting is prolonged, the proteins stored in the liver will also participate in the production of glucose. If the fasting is prolonged even more, fat begins to be mobilized. The fatty acids from this fat go to tissues that can use them as energy source, such as the heart and muscle, while the glycerol goes to the liver where it also participates in the production of glucose. Low blood glucose levels are also detected by the pancreas, which in response produces a hormone called glucagon that has a function opposite to that of insulin. Glucagon facilitates the mobilization of stored biomolecules, especially glucose. If the fasting is prolonged beyond 12 hours, stored glucose and proteins are reduced as well as stored fat. In this situation, the mobilization of fat increases. Glycerol from this fat continues to make its way to the liver to participate in glucose generation, and fatty acids continue to make their way to organs and tissues that can use them as an energy source. But in addition, fatty acids also go to the liver where they participate in the production of ketones. Ketones can pass into the blood and go to organs and tissues that can use them as a source of energy, such as the muscle and the heart, but also the brain. This type of fasting results in reduction in fat, and therefore an anti-inflammatory effect is attributed to it since fat is considered pro-inflammatory. In addition, ketones in the brain seem to be neuroprotective, so prolonged fasting is also considered neuroprotective. If the fasting is prolonged for a very long time, the reserves of glucose, amino acids, fatty acids, and glycerol can be depleted, leading us to a state known as starvation. In this state, cells cannot obtain the biomolecules they need for their functioning and survival, so they end up dying. Under these conditions, cells begin to digest parts of themselves in order to obtain the biomolecules they need to survive in a process known as autophagy. Cells can digest their own cell organelles by fusing them with vesicles called lysosomes that are loaded with enzymes. This type of autophagy is called macroautophagy. Cells can also digest other molecules in a process called microautophagy, or digest molecules, which must first bind to proteins called chaperones in order to enter lysosomes. In this case, we speak of chaperone-mediated autophagy. Once inside the lysosomes, both the organelles and the molecules are digested by the enzymes present in the lysosomes. The final result of the three types of autophagy is the generation of biomolecules that the cell can use to obtain energy or to synthesize structures that it needs for its survival. Autophagy is not only important because it allows combating starvation, but also seems to be beneficial in eliminating pathogens, in degrading damaged proteins, which are very important in neurodegenerative diseases, but also during elimination of damaged cells of tumors. Thinking about the possible beneficial role of autophagy, there are people who fast with the idea of inducing autophagy, although inducing autophagy is not so simple since we do not know in humans how long a fasting period has to last to induce it, since autophagy is intended for borderline starvation situations. In mice, it has been demonstrated that a 48-hour fasting is capable of inducing autophagy. However, it is important to note that after 48 hours of fasting, mice lose 20% of their weight, which is a lot. In fact, after 48 hours of fasting, when mice induce autophagy, they are in a precarious situation, and a few more hours of fasting would be lethal. In humans, we do not know how many days of fasting are necessary to induce autophagy. Humans only lose 2% of their weight in 4 or 5 days of fasting, which suggests that very long fasting is probably necessary to induce autophagy. More studies are required to know what type of fasting is capable of inducing autophagy in humans.